All right, so we are starting the second day. Welcome to the Thermo Fisher Science Scientific Forum. So we'll start with a welcome address by our Dean, Dr. Alex Stegnaro Green, Regional Dean of the College of Medicine here at the College of UIC. So, Alex.
Well, good afternoon. Thank you for the introduction, Dr. Ramaswamy. This is the 42nd Summer Science Program co-hosted by the University of Illinois College of Medicine, Rockford, and Thermo Fisher. I want to welcome not only those of you who are here with us in the auditorium today, but those who couldn't join, either because you couldn't make it or because of the limitations secondary to COVID-19. And we appreciate everyone being so adaptable and being able to move forward, not only with the program today, but more importantly, the program over the summertime. Just to tell you a little bit about the University of Illinois College of Medicine, we are the second largest college of medicine in the country. The largest one, by the way, is Indiana University. We have campuses in Chicago, Peoria, Urbana, and Rockford, and we have about 320 medical students that we train a year, hundreds of graduates and master's students, and hundreds, if not even in the thousands, of residents and fellows. So it's a very, very large educational institution. And one of our core components is research. And that's what we're here to celebrate today and to talk about. So what's the goal of research? Why do we have research as an integral component of an academic health center? And there are two reasons. One is to promote science, to improve the health and well-being of patients, to prevent disease, to cure disease, to alleviate suffering, and to address all the problems that we have as individuals in this world. So here at Rockford, we are doing research in a whole variety of areas, and some of which our 8 students in front of me, the high school and college students, were involved in this summer. We have research going on in Dr. Ramaswamy's lab on vaccine development for lymphatic filariasis, elephantiasis, and a dog vaccine for heartworm. We have work in Dr. Lee's lab, and I think Dr. Divya's lab, in nanomedicine, stem cell generation, Dr. Matthew, biotribal corrosion. And what's really exciting, I think, for the 8 students in front of us is you got to be involved in cutting-edge research. Doesn't matter where you would go around the world, you would not be involved in research that's more cutting edge, that's better funded, that's better published, that's better respected than the experience you have here. And the second reason why we do science is not only to move things forward in terms of improving health and well-being, but it's also to train the next generation of scientists. So although it's hard for you to think it right now, the 8 of you one day are going to be the next generation of scientists. And those of us in here will be finishing up our careers and reading about all the incredible things you're doing and the new technology and how you advance science and moves things forward. And I want to take a second to thank all of our faculty, all whom I mentioned and those who I haven't had a chance to mention yet. But we could only run programs like this because of the incredible dedication of the faculty of the Department of Biomedical Sciences, who see it as integral to their mission of being in an academic health center, training the next generation of scientists, be it those of you who've been in the summer program, those of you who are in the MBT program, doctoral students, postdoctoral students. It's part of that continuum that makes us also proud to be parts of academic health science centers. And finally, I'd like to conclude by thanking a number of individuals. I want to thank Dr. Ramaswamy. It starts from the top. He has an incredible dedication to science and training the next generation and mentoring individuals, the faculty that you work with, who I've talked about, all of the support staff that make it possible, and finally, for Thermo Fisher, without which we would not have had a program for the last 42 years, whose support and understanding and appreciation and dedication to training the next generation is so transparent in everything we do. So welcome, and I'm going to turn things back to Dr. Ramaswamy.
Thank you, Dr. Stegnar-Green. So Dr. Stegnar-Green talked about a little bit about the college, what we do. Over the summer, one of the things which I wanted to echo what Dr. Stegnar-Green mentioned is about the next generation of scientists. I mean, even though it is a short period of time that you have been here at the College of Medicine, that 8 weeks, or at the Thermo Fisher Scientific, the 8 weeks or 10 weeks opened up an avenue for you for the future. I know several of the students wanted to come back into the medical field, and we see some of the future doctors sitting here, and also some of you really wanted to go into the PhD program, and also some of you in the engineering field. So this is probably one of the smallest experience that you have, probably you'll carry long in your career, and you'll remember this experience, what you had here. So the training happened both at the Thermo Fisher Scientific and also here at the College of Medicine within the Department of Biomedical Sciences. So the Department of Biomedical Sciences, we conduct all the basic research in this campus. We have researchers working in several aspects of clinical medicine, basic research into that one leading to translational medicine. So cancer research done by Dr. Neil Puri. I know that we have one student work there. And also we have Dr. Gnanashekhar, and we have students work under him. Then, of course, we have Dr. Matthew, who works on mechanical engineering, but work on engineering aspect of the medicine, and Dr. Lee working on stem cell. And then we have Dr. Aoshang Chen and Dr. Goshing working on autoimmune diseases. Then, of course, Dr. Divya Vijayakumar, she works on nanotechnology, trying to develop new medicine for some of the heart to get some treatment. Then finally, my lab works on vaccine research. So the major source of funding for all these comes from NIH, the National Institute of Health, that supports our research. Especially if you don't have funding, then probably the research is impossible to do. Second, we also get funding from certain foundations that support this research, National Science Foundation and also other private foundations that support the research. One of the major foundations at Rockford that supported the research is Blazer Foundation of Rockford. So they supported us in establishing the RMDR lab, which is the regenerative medicine and disability lab, the nanotechnology lab, and also they recently, this year, they funded us $500,000 to establish a 3D bioprinting lab. So we have a 3D bioprinter right now. It's all set up, and I think Dr. Vishal Khatri, he's actually the director of that lab, and he's setting up starting. So I know that Thermo Fisher was interested. There is other universities, including NAU, was interested in working with us to print some of the tissues work. As I said yesterday, in the future, in another 10, 15 years, NIH will want to reduce the number of use of experimental animals, mainly the mice and rats. So they want the research to be done in human or human tissues, where the 3D printer will be the new generation that's going to be future. So I think we are probably about 15, 20 years ahead of time, and this is a wonderful opportunity for our faculty and also for the next generation scientists that will be trained here starting next year. The department also offers a master's biotechnology program. This was started in 2018. We have graduated several students, and all of them have employed 100%. And we recruit anywhere from 21 to 30 students each year, come back to the program and get trained within 2 years. They finish and get employed. So with that brief introduction about the department, I would like to invite Dr. John Rogers. He is the director of the research and development at Thermo Fisher Scientific. I invite him to give a brief background about the Thermo Fisher Scientific. John. So this will advance the slide.
Very good. Down. All right. Well, I want to thank everybody for this opportunity to serve as a little bit of a spokesperson for Thermo Scientific. It's great to be back in Rockford. A year ago, I moved from the Rockford area to Oregon. This is my first time back, and it's a great honor to come back and spend some time with you. I grew up in the Rockford area, went to high school at Harlem, and back in 2007, after my undergraduate, graduate, and graduate degrees and some other work, I joined the Pierce site, the Thermo Scientific site in Rockford here. And so it's really an honor to come back and spend some time with you. As was said, you know, we celebrate 42 years of collaboration between Thermo Scientific and the School of Medicine. We've had over 540 interns in the program, both high school and college. And really, this summer, especially as I've seen the presentations and reviewed the abstracts, I'm so impressed by not only the technical expertise that's being developed, but also the, maybe it's competitive, maybe it's just passion. You know, I love passion. I love it when people care about what they do. And what's great about this program is that it inspires people. It brings that passion. And so I really applaud all the participants in the program, the judges, the mentors, the labs, for bringing passion to science. So this year we had 5 high school and 4 college interns complete the training. And I want to spend just a little bit of time talking about the context here. So as I said, I grew up in the Rockford area. I now manage R&D efforts across teams here in Rockford, as well as in Eugene, Oregon, and in Carlsbad, California. But all starts here. My undergrad was in biochemistry and computer science, and I was always intrigued by this idea of combining different ideas, chemistry, biology, and computer science. And so, you know, coming back to share some time with you, it always inspires me with new ideas about what we might do. Yep. So, as Thermo Fisher Scientific, as an employee, I want to spend just a few moments talking about the company. So, Thermo Scientific is a very large company, over 80,000 employees across 50 countries. We generate over $30 billion of revenue a year. And we invest more than $1 billion a year in research and development efforts here in Rockford and at other sites around the world. It's an honor and a privilege and a responsibility. And we offer unparalleled commercial reach. So we reach customers around the world every day. Now, you know, as far as unmatched depth, we develop and commercialize innovative technologies, whether that's through internal research and development efforts to create new products, whether it's working with academic partners to commercialize some of their ideas, turn it into something that everybody can use, or working with small companies that have some great ideas and are looking for a partner to make them available to the world. It's really an honor. I think, you know, between my groups, we work with more than 80 labs around the country on collaborative projects to look at new concepts for product development, to evaluate new ideas, and to apply our products in novel ways. And so it's a great opportunity to not only change science and advance science, but also to better understand how our products can be used in new ways. We work as a partner with labs, and you know, various aspects of Thermo Fisher Scientific play different roles in how we help advance science. We have a number of premier brands, so many people are familiar with Thermo Scientific and Fisher Scientific, as well as the Applied Biosystems, the Invitrogen brand, the Unity Lab Services, and for pharma customers, the Patheon products, Patheon line for biopharma production support. So, you know, I love our mission statement. You know, people look at it and sometimes say, well, that's really squishy. I don't know. But what I love about it is that every day we go to work and we walk the talk. Our mission is to enable customers to make the world healthier, cleaner, and safer. And I challenge you to talk to anybody at our site that doesn't know this mission and know how they impact it. I think it's so extraordinarily important to know how your efforts can change the world. And it just brings passion to me. It's inspiring. And so, every day we think about how we can make the world a better place. And we certainly have a lot of openings, so I want to encourage you to think about that. Here at the Rockford site, we are experts in protein biology. Across all of Thermo, we're recognized as the center of excellence for protein biology. We develop kits, reagents, and services that enable researchers to better understand proteins, whether it's for vaccine development, whether it's for therapeutic drug compound screening, whether it's for biomarker discovery, whether it's for a better understanding biology and what's happening in different disease states or under different conditions. We sell more than 40,000 products out of Rockford. We handle over 1,000 orders a day. So at the end of the day, there's a lot of boxes moving through the shipping area. We sell in 79 countries. And it all started with Alan Pierce back in 1950, buying some land over on the west side of Rockford and extracting chlorophyll from alfalfa. That's pretty impressive. So I have a lot of pride in Thermo, and it's really an honor to share some of that, what I feel about Thermo with you. So, in more context, this COVID epidemic, this pandemic, has had a tremendous effect on our business. We've had people that had to split shifts, had to work from home, had to initially respond to what's going to happen to my job, and it turned into how can we crank it up? So we make essential lab equipment and personal protection equipment, your masks, your gloves, your COVID test kits, the Q-tips. We help discover and understand ways to treat this disease, to detect it and treat it. So my colleagues at the Eugene site, as an example, we had to scale up some of our dye development for these PCR assays over 1,000-fold because the intent and the delivery was we wanted to be able to support more than 10 million assays a week around the world. And we did that. But it was not without a lot of effort. We offered tools not only to discover, quantify, and validate vaccines and treatments, but we also offer complete testing solutions. So many of the COVID tests that you're getting results from today are coming from Thermo Fisher Scientific Instruments and reagents and assays. We've also worked not only with PCR instruments and DNA sequencers, but used cryo-electron microscopy to better understand the structure of the virus and how to design better vaccines. And so, more recently, we assist a lot of partners in the manufacture and production of vaccines for customers around the world. So definitely bringing this idea of cleaner, healthier, safer to the world every day. We also help on a community side. So we've provided, I think, something like $19 million in support for needy countries, for response to the pandemic. We've provided significant numbers of millions of kits for countries around the world that need to know the level of the pandemic. So besides the pandemic response, we have a lot of local support. So we provide support locally, over $1 million a year, to local organizations. We also have STEM programs and community action council activities that connect our research and our site with efforts around the community, whether it's through science or whether it's through helping with a food bank or other community contributions. Now, one of the things that I wanted to highlight here is, with the success of this internship program is certainly in developing new scientists. But for myself, as a leader, it's also extremely valuable for us to develop new leaders. So, there's always a reciprocal interaction here. When we bring in a summer intern, we find a mentor that's going to take responsibility for ensuring that that intern is successful. And that's a great opportunity and great responsibility. And I really appreciate when people step up and say, I'll take an intern because I know it's going to take some time, but it's my opportunity to teach somebody else, to get a new perspective on the work that I do and how it can impact others, to try out a new idea, to try out a new application, to learn more about myself and how I can work better with others. And so I see it as it's a win-win opportunity. We win because interns learn more about science, but we also win because future leaders learn more about how to work with people and how to manage and how to help them succeed. That's part of leadership. So with that, I think I'm going to hand over to Janaki to introduce the presenters. And again, I want to thank you for this opportunity, and I certainly look forward to some great talks.
Thank you, John. That was really a nice introduction, and you brought out very important points, whether it's about the passion of the students or the desire of the mentors to develop themselves and others. It was great to hear that. Thank you. So to start the student presentations, I want to just take a step back and talk about what this day is about, the Summer Science Forum. I think we're going through some slides here. Can we? We get a second look at it, so that's good, right? Can I have, yeah, thank you. Okay, so Summer Science Forum, what is it? Today is the day that we celebrate the end of internship. So all the students who have put in hard work over previous weeks, last 8 to 12 weeks, they get a chance to present their projects. But for them, today, actually it started very early in the morning because today is also a competition for them. They had an interview with a panel of 5 judges. The judges are seated over that side. And where they got to present their project for 3 minutes, and then they were quizzed for nearly 7 minutes. So it was a 10 minutes per student interview. And the goal of this interview is to see that how well the students understand their project, how well can they communicate about their work that they have done. It is really important for us to know that we are training them in the right way. They are getting the most benefit that they can. And if they go outside, they'll be able to represent the information correctly. So it's part of the training. They will, in future, need to go through the interviews. So this is an excellent training for them. So right now, we are in the second phase, the afternoon phase. Here, the students get to present the data, or the presentations. Each one will get a chance to talk for 10 minutes. And for the college students, there are 3 undergrad students over here, we will have a question and answer session after each of the presentations. The high school students will not be having the Q&A session. So the rules for the presentation, like I mentioned, each student gets 10 minutes to talk on their work. At 9 minutes time, you will see a timekeeper who's seated in the center. She will raise an orange flag just to let you know that you're nearing completion of your allocated time. At 10 minutes, which is the allocated time, she will raise a red flag or red paper. So at that time, I request the students to make sure that they are wrapping up their presentations because we don't want to go too beyond that. So after each, as I mentioned, the high school students, after they're done, they'll exit. For our undergrad students, we'll have the Q&A session. So to kick off today's presentations, the first speaker will be Ruth. She is a high school student. She's going to be a junior this fall at Auburn High. She wants to become a doctor, but she also wants to keep in touch with the research. So she's going to be most likely MD, PhD, or that would be her goal. Her hobbies and interests include reading realistic fiction novels. So I'll turn it over to Ruth for her presentation.
Can you hear me? So good afternoon. Thank you, Dr. Janicki, for the introduction. Today, I'd like to talk about investigating hereditary spastic paraplegia axonal degeneration by characterizing SPG15-induced pluripotent stem cells. So to begin with, hereditary spastic paraplegia, or HSP, is an inherited group of neurodegenerative diseases that is characterized by progressive stiffening of muscles throughout the body, particularly muscles in the lower limbs and the hip muscles, due to axonal degeneration of cortical projection neurons. There are around 83 subtypes of HSP, including SPG15, which is the variant that my study focuses on, and the prevalence is around 2 per 100,000 worldwide. So it's quite a rare disease. However, it can have a drastic impact on the lives of those who are diagnosed with it, in the most extreme of cases, completely immobilizing patients. So there are various common sociological HSP impairments, but one of the most recent findings has been this critical connection between mitochondrial dysfunction and axonal degeneration in neural cells. So the cause of this mitochondrial dysfunction most often is an imbalance between mitochondrial fission and fusion processes in the body. And this imbalance can lead to axonal degeneration due to the inability of mitochondria to mobilize properly, as shown in the video in the screen, along the axons, as well as perform the vital functions that it does in cells, such as regulating redox signaling and ATP production. Some visible axonal defects that are caused by this mitochondrial dysfunction include reduced axonal outgrowth and cell apoptosis. So spastic paraplegia type 15, or SPG15, is the most common recessive variant that represents 2 to 4% of HSP cases. It is caused by genetic mutations affecting the ZFY E26 genes that code for the protein spasticin. So spasticin is a protein that regulates lysosomal functions within cells. So when there is a genetic mutation that inhibits spasticin from performing its function properly, this can lead to abnormal cell autophagy, or the breakdown of cells, thereby resulting in impaired mitophagy with regards to mitochondrial dysfunction. And ultimately, this can result in axonal degeneration and various neurodegenerative diseases, one example being SPG15. So the current problem with HSP SPG treatments, which involves various physical exercises and use of oral antispasmodics, is that they have not proven to reverse or even significantly inhibit HSP symptoms. So therefore, this study aims to utilize and test for the pluripotency of SPG15-induced pluripotent stem cells, or iPS cells, that have been derived from patient-specific fibroblasts, serving as a first crucial step to creating a stem cell model that can later be used to test for inhibitors of axonal degeneration. Before I go into my hypothesis, I'd like to talk a little bit about how we can confirm the pluripotency of induced pluripotent cells. So once iPS cells have been derived from corresponding somatic cells, it is critical that they be tested for their ability to differentiate into specific cell types, in this case, neural cells. And one of the most common ways to test for cell pluripotency is to analyze the expression of certain pluripotent transcription factors, as well as certain pluripotent proteins. One example is SSEA4, which is a stem cell surface marker. So therefore, in this study, we hypothesized that the reprogrammed SPG15 iPS cells will indeed express certain genes and proteins that indicate cell pluripotency. In order to test our hypothesis, our aim was to characterize the SPG15 iPS cells, and as part of our aim, we had 3 tasks. The first task was to actually generate the iPS cells by reprogramming fibroblast cells using the 4 transcription factors. The second task was to test for and compare the genetic expression of pluripotent transcription factors between our original fibroblast cell sample and our generated iPS cell sample, using both regular and qPCR as well as gel electrophoresis. And finally, our third task was to analyze protein expression in our generated iPS cells using the process of immunostaining. So for task 1, once we collected our patient-specific fibroblasts, we utilized Sendai viruses that contain vectors with the 4 reprogramming factors, OCT3-4, SOX2, KIF4, and LMIC, in order to transduce our fibroblasts. Once the fibroblasts were transduced, they were then cultured in fibroblast culture media for around 6 days. Then they were passaged onto mouse embryonic fibroblasts and cultured in human embryonic stem cell. And after 24 days, we can see that the iPS colonies begin to form in solution. So for task 2, in order to begin testing for a genetic expression of transcription factors, the RNA of the iPS cells was isolated and then reversely transcribed into cDNA using the enzyme reverse transcriptase. The cDNA was then mixed with GOTAC and the corresponding primers, depending on what gene was being tested for, in order to run and create our PCR mixture. The PCR was run. We conducted gel electrophoresis in order to separate the amplified gene segments. We used a gel imager in order to observe the genetic expression. So first, we tested for the expression of our housekeeping gene DAPT, and we were looking for there to be approximately equal expression in both samples, both our fibroblasts and our iPS cell sample. And the concentration of cDNA that was used to establish that result was then used to test for the expression of other genes that uses a similar workflow. So we can see here from our results that the gene DAPT had around equal expression in both our fibroblast sample as well as our SPG15 iPS sample, indicating that a sufficient concentration of cDNA has been established to begin testing for the expression of other genes. So the gel at the top represents the expression of the gene FGF5, and we can see that the fibroblasts had a much greater expression of that gene than the iPS cells. Conversely, the gel at the bottom shows the expression of the 3 pluripotent transcription factors, OCT4, NANOG, and SOX2. And in all 3 cases, the iPS cells had a much greater expression of those pluripotent transcription factors than the fibroblasts, which had almost no expression, thereby genetically validating the pluripotency of our derived iPS cells. But in order to take it one step further, we wanted to confirm our results using qPCR. So in order to create our master mix, we mixed together cyber green, purified water, and our primers. Then we pipetted 60 microliters of our master mix into each of the wells that was being used, as well as 4 microliters of the corresponding fibroblast and SPG15 cDNA samples. We sealed and centrifuged our qPCR well plate before running our qPCR. So we collected and averaged our CT value data, which we then used to calculate our fold difference. And the fold difference is the value that we use in order to compare genetic expression between our untreated sample, our fibroblasts, as well as our treated sample, our SPG15 iPS cells. And so from our results, we can see that the SPG15 iPS cells had a significantly lower expression of the gene FGF5 than our original fibroblast sample, thereby helping us confirm cell pluripotency, as the SPG15 iPS cells do not significantly express a gene that would otherwise indicate a lack of development from our original fibroblast cells. And finally, for task 3, in order to analyze protein expression within our generated iPS cells, we use the process of immunostaining. And in immunostaining, we utilize antibodies in order to visualize proteins of interest under a microscope, fluorescent microscope. So in our procedure, we first fixated our SPG15 iPS cells onto cover slips using 4% paraformaldehyde. Then we used 0.2% tritone in order to slightly break up the cellular membrane to make the proteins accessible to the antibodies for binding. We added donkey serum to block nonspecific binding. We then added our primary and our secondary antibodies, the secondary antibody housing the fluorochrome that would make the proteins visible under the fluorescent microscope. We finally added our Hoche dye and mounted our cover slips onto our slides. So these are the images we were able to take of the iPS cells under the fluorescent microscope. The Hoche dye in blue represents the cellular nucleus of our iPS cells. The OCT4, NANOG, and SOX2, they're all proteins that indicate cell pluripotency, and the SSEA4 in red is a stem cell surface marker. So the very evident and defined presence of these pluripotent proteins within our SPG15 iPS cell sample helps us to once again confirm the pluripotency of our iPS cells. So as a discussion, from our genetic testing, we were able to see how the SPG15 iPS cells had a much greater genetic expression of the transcription factors OCT4, NANOG, and SOX2, whereas the patient-specific fibroblasts had a greater expression of the gene FGF5. And from utilizing immunostaining, we were able to see how our iPS cells had a clear expression of our pluripotent proteins OCT4, SOX2, NANOG, and SSEA4. And these results collectively help us confirm the pluripotency of the SPG15 iPS cells, and thereby their ability to differentiate into various neural cells, for example, the cortical motor neurons presented at the top right of the slide. So in conclusion, our hypothesis was validated as we were able to generate SPG15 iPS cells whose cell pluripotency was confirmed using our genetic testing of pluripotent transcription factors, as well as by analyzing the presence of pluripotent proteins. And this result is actually quite critical to current hereditary spastic paraplegia research, as now we have a validated pluripotent stem cell model that can be differentiated into SPG15 cortical motor neurons to be characterized and experimented on with various inhibitors that directly target axonal degeneration, rather than continuing to rely on current treatments that do not effectively address the symptoms at hand. I would like to acknowledge everybody on the side, particularly my mentors, Dr. Lee and Dr. Mel, as well as Thermo Fisher Scientific and the College of Illinois, University of Illinois College of Medicine for this wonderful opportunity. Thank you.
One thing that I forgot to mention when I was introducing Ruth, she did acknowledge her mentor. She, over the summer, worked in Dr. Lee's lab. One request that we had made to all the mentors was to give us a sentence about how they would describe their intern, and Dr. Lee said that Ruth was very smart and diligent worker. So thank you, Ruth. So our next presenter, there we go. She'll be a college graduate next year. We'll have Anna as our next presenter. Anna is going to be a senior this fall when she returns to NIU. She's majoring in chemistry with a minor in biology. She wants to pursue a career in medical biotechnology. And when she gets a free time, she loves to sing, hike, and participate in photography or take some pictures. Over the summer, Anna worked with Suzanne Smith. And Suzanne describes Anna as a person full of scientific curiosity. So Anna.
Good? Okay. Hello, everyone. My name is Anna, and I'm excited to share with you the project that I was working on this summer, which was the optimization of protein purification from XP293 and XP-CHO expression systems through the use of Pierce high-capacity protein A mag beads that are alkali stable. Oops. So mammalian cell culture is really key for biotherapeutic productivity in the fact that we need to be able to have a way to screen different types of biologics, such as antibodies and therapeutic proteins, in order to determine their efficacy as therapeutics. And so over here, we see that the XP expression systems have been modified in order to withstand high-density cell culture and also to express a high quantity of proteins through the use of transfection reagents that are able to enhance and feed the cultures. So I maintained these two different cell cultures this summer for the purpose of developing the expression and purification optimizations for 3 different FC fusion therapeutic targets. Our workflow is seen here, starting with the suspension cell culture of our 2 cell lines. And the XP293 is a human kidney cell line, and the CHO is a Chinese hamster ovary cell line. So once we were able to collect the targets that we were needing, we used DNA constructs in order to express rilonacept, abatacept, and alefacept as our 3 targets. And then once these were secreted into the media, we optimized the protein purification system using a Kingfisher Apex benchtop instrument. We then separated the fractions using SDS polyacrylamide gel electrophoresis in order to analyze the proteins by their molecular weight. And finally, we quantified the elutions using a nanodrop 1 instrument, which measures the absorbance of the proteins at 280 nanometers. And so this was a UV-Vis spectrophotometer. So the 3 targets that we were studying are commercially available as treatments for conditions such as pericarditis, rheumatoid arthritis, and plaque psoriasis. And the structure of the proteins, as seen here, is similar to an immunoglobulin that you see on the left. The structure that was especially interesting for us was the FC fragment crystallized domain, which is what can bind to the protein A of our magnetic beads for the purification process. So once we had expressed our proteins of interest and they were secreted into the media, we harvested them with 3 different methods. The first clarification method that's traditionally used takes a 2-step process of centrifugation followed by filtration. The second, which is spin clarification, is a shorter process and skips the filtration step. And the unclarified was the especially interesting one for us to determine whether we can purify protein directly from the cell culture. So the support that we used for our affinity chromatography purification was the protein A mag beads, as mentioned earlier, which is constructed from agarose that has been embedded with ferric oxides. And so the protein A ligand is what can bind to the proteins of interest and carry them through the process. So once you have added these beads to a supernatant and the binding affinity has occurred, you can place those on a magnetic stand as seen here on the lower right, and then you can pipette away any of that supernatant, and what's left behind is the target of interest on your mag beads. The beads that I used in my project were recombinant protein A mag beads that were from the construction of N-hydroxysuccinimide functional groups that reacted with the primary amines of protein A in order to form a covalent bond between the two. And the FC fusion proteins were then collected from the protein, the FC fusion targets were collected from our cell cultures by the bond that was formed between the protein A and the FC domains. We used a Kingfisher Apex affinity purification system, and so the way that this works is that we use 96 well plates, and these plates each represent a step in the purification process. And so the way that the instrument works is that it will actually transfer the beads from the plate to the next plate throughout each of the steps. And so we can see here at the bottom for our entire workflow. So we started with the 3 different clarification methods, and then we allowed the beads to incubate with those start samples for an hour at room temperature. We then, once we removed the beads, what was left behind is considered the flow through, and it's a measure of how well the binding capacity was able to be determined. And so this is the part that really needed to be optimized in order so that we didn't have any of the protein of interest being lost in the process. And so the 3 wash steps that followed, we used phosphate buffered saline as well as T20 detergent in order to eliminate some non-specific binding elements. And finally, the elution was performed using glycine, which effectively removes the targets from the beads. And then in order to regain a physiological pH, we used tris and at pH 8. And then finally, the last step of the bead boil used diethyl-threotol as a reducing agent, which then reduced or removed any extra elements that may be sticking to the beads in order to determine the purity of our products at the end. And this was using harsher conditions. So we took the fractions from our purification and then ran them on our gels, as you can see here. So we have our 2 cell lines and our 3 clarification methods. And the main goal is to check to see if the unclarified starting samples have a similar purity to those that went through the clarification process on the far left. And so we do see that the elutions came out very clean, and we can see that this is a very efficient method for saving time for high throughput screenings and purifications. But we also had 2 more targets to investigate. So for the abatacept, we are again looking to see if the clarification is consistent across these methods. And we do see that there is plenty coming off in the wash steps in order for us to determine that there is a clear elution in the clear purity and high purity in the elutions from the unclarified starting samples. And finally, with the Alephecept, we're again looking for a similar pattern. And this is what we do see with our 2 cell lines. And so the final conclusion is, again, that this process works additionally well for the 3rd target. So we then quantified the elutions to determine what sort of amount of protein we could recover. And we were expecting to see a similar pattern that we saw with the SDS page analysis. And so as we can see, the unclarified does exhibit a slightly higher elution quantity due to the fact that there are trace amounts of other proteins that may be carried along. But overall, it does follow a similar trend as we had seen in our gels. And so we also can tell that there's difference in the expression of cell lines because the Verlanocept and Abatacept are more highly expressed in the CHO cell line versus the Alephecept, which is more highly expressed in the 293. And again, this was just shown in one example of a purification, which is why the statistical analysis is not included here. But this is showing to be effective in our purification process. So in summary, we can see that we were able to successfully purify and express our 3 targets of interest from our 2 cell lines. And we were able to compare the starting sample prep in order to determine that there was definitely a high amount of purity that was resolved from our unclarified starting samples. This was due to the high binding capacity of our protein A mag beads. And this is, again, a relevant application in biopharmaceutical industries in order to save time and have a high efficient way to screen many different types of therapeutics. This project was due to a great teamwork. I'd like to thank my mentor, Suzanne Smith, for all the time and training that she's invested, as well as the wonderful staff at Thermo Fisher Scientific for a wonderful internship this summer. And with that, I thank you for your attention, and I welcome any questions that you may have.
There's a question on the side. No? Oh, okay. I thought you raised your hand.
So, the cell culture system is a very simple system actually to do anything, but you are planning to do something on a patient, or right? That's the long-term goal of this study, right?
Right, so we can determine from studies like this where there is a way to purify targets for therapeutic purposes and save a lot of time and be able to do this in an efficient way with high throughput screening.
So did you ever try like any kind of sample of blood or urine or something? Did you ever try that out?
In this case, we used these 3 specific targets, but we could definitely expand it into those types of investigations as well.
And then, did you do a mass spec study with the, with the eluent to see if you do? Because I did see one of your cell samples did have another band in them. So did you? I mean, it's there is another band which I can see in the sample. So what is the level of purity? Because when right now cell lysates are very simple, but when you come to patients like blood and other things, it's a lot of complexity. So do you think that you would? What is the level of purity you would think you would get?
That's a good question, yes. So that is true. We were not able to use mass spectrometry in order to verify all of the components as we would expect. But in this case, we were able to see that according to the gels, there would be about 90 to 95% purity from our starting samples. But that is, again, a good direction to head for further analysis.
There are a couple of questions on this side.
So you were using magnetic beads and automation for most of these purifications. Did you do any of these processes by hand? And can you talk to the importance or relevance of that, of the automation versus other methods?
Yes, I did do the manual method as well. And so in comparing the two, there was sometimes a little bit more elution that we could achieve through a manual system. But with a high throughput type of screening where you need to have multiple targets purified all in one batch, the automated system was definitely a time saver in that case. And also in terms of optimizing a purification, it really saved a lot of time in terms of the amount of different tests that you could run at the same time to quickly analyze which method works best for this type of purification.
In addition to the work you were working on, did you and your mentor discuss other applications that instrumentation is used for, like the Kingfisher?
The Kingfisher, in terms of what it can be used for other types of applications, yes, there's a lot of different types of applications, including analyzing things for mass spectrometry, sample prep, and so that's just one facet, but there's a lot of different applications. The Kingfisher instrument has also been used for spike protein or COVID analysis for testing many different types of diseases, but that's one that was definitely recently used for a very high volume of samples.
Yeah, Dr. Ramaswamy.
Can you use this automated technology for any protein, recombinant proteins?
Can it be used for any protein? That's a great question. I think it depends on what sort of ligand will bind to your proteins of interest. And so there would be an interesting direction to analyze which type of resins or beads may be able to bind best to your targets.
So is that specific to only the FC region?
In this case, we were studying the FC domains, so different structures that have that particular component or that domain would be applicable to any type of protein A purifications.
Thank you, Anna.
Thank you.
So our next presenter will be Varun. Varun is a high school student. He worked over summer in Dr. Chen's lab. When he goes back to school, Varun will be going back as a senior at Auburn High. He would like to pursue a career in medicine. His interests and hobbies include playing basketball and tennis and having fun with his friends. Dr. Chen describes Varun as a person who is very enthusiastic and intelligent student researcher with an innate interest in science, and he's a delight to have in the lab. Varun?
Can you guys hear? All right, so this summer I worked on developing a more effective immunosuppressive vaccine for atherosclerosis. So atherosclerosis is the underlying cause of the #1 killer in the United States, which is heart disease. And the deaths due to heart disease have been increasing over the past decade and have reached an all-time high of 700,000 deaths in the United States alone as of 2020. And these problems are associated with the adoption of a Western-type diet, a high-fat diet that Americans have been consuming. So atherosclerosis is the significant buildup of plaque in the arteries released to the narrowing of the arteries. So as you can see here in this first picture, you have a normal artery with proper blood flow, but when plaque actually builds up, the artery becomes slightly narrowed and the blood flow is slightly restricted. And you can see here, when the plaque actually ruptures, the platelet cells will come to stick to this rupture and cause a blood clot, which can restrict the blood flow. However, most deaths actually occur when this lesion ruptures and the debris spreads to other places via the arteries and causes instant blood clots, which can cause strokes and heart attacks. So knowing that the plaque is detrimental to the body, we need to know what actually composes the plaque. So the endothelium is a thin layer of cells that lines the arteries and provides its protection. So when damaged, low-density lipoprotein, or LDL cholesterol, can penetrate the endothelium and start to accumulate in the arteries. So some factors that can lead to unhealthy endothelium are hypertension, low shear stress, systemic inflammation, diabetes, and genetic determinants. So inflammation is the key player in the immune response in atherosclerotic lesions. So when your lesion actually ruptures, these platelet cells will come to stick, and the inflammatory cells will come to stick, which causes the blood clot to form, which can kill you. So you have your LDL cholesterol, and this will eventually become oxidized, and these macrophage cells will actually engulf the oxidized LDL. From there, these will become foam cells, and an antigen-presenting cell or dendritic cell will come in contact with this foam cell and allow it to present itself to these naive T cells. So there are many key antigens in atherosclerosis, like ApoB, HSP60, and many others. But we believe that ApoB is the key antigen in driving inflammation. So this dendritic cell will actually present the ApoB antigen to the naive T cell, and from there, the naive T cells are able to produce these T effector cells, which drive the inflammation process. So they directly produce pro-inflammatory cytokines, which drive inflammation directly. And this will be an ongoing cycle, on and on, which drives inflammation, makes it very detrimental to the body. So some current treatments for atherosclerosis. So you have bypass surgeries and stents. And stents as shown here. And these actually, stents hold open the lesions to allow for proper blood flow. And you have cholesterol-lowering drugs. And looking more at statins, they pose a long-term toxicity to the body, and they inhibit systemic inflammation, which is not a good thing because inflammation is the key player in regulating the healing when you have any sort of injury or disease or virus. But however, they do not inhibit inflammation at the lesion site in atherosclerosis, which is problematic. So knowing this, we need an alternative treatment that actually reduces inflammation specific to atherosclerosis at the lesion site. So normally in the aorta, in the arteries, you're going to have effector T cells, which stimulate the immune response, and regulatory T cells, which actually suppress the immune response. So normally in the aorta, you have more effector T cells than regulatory T cells, meaning that whenever you have this rupture in plaque, there will actually be inflammation. So the purpose of our vaccine is actually to raise Treg levels, and by doing so, it'll actually shift the balance. And this, therefore, will actually induce anti-inflammation by targeting the ApoB atheroantigen. So our hypothesis is that by immunization, raising atheroantigen-specific Treg may reduce atherosclerosis. We can analyze this by determining whether or not immunization actually reduces atherosclerosis as assessed by our first aim, end-phase analysis, and our second aim, morphometric analysis. So our suppressed immunization is our vaccine, or 3208, and this is the vaccine that raises Treg levels to induce anti-inflammation. So it's composed of 2 proprietary adjuvants, 3 micrograms of one and 20 micrograms of another, plus a third adjuvant, dexamethasone, which is 8 micrograms. And these serve as our anti-inflammatory drug that can be delivered. And we target the ApoB atheroantigen because we believe that this is the key player in the immune response. So normally the ApoB antigen will be presented to the naive T cell to produce effector T cells. But when you overload it with the anti-inflammatory adjuvants, it'll actually produce T regulatory cells instead to induce anti-inflammation in the lesion. So now we want to test our hypothesis through an atherosclerosis mouse model. So all of our mice have been genetically engineered to be ApoE knockout mice. So if you knock out the ApoE gene, cholesterol won't be circulated back to the liver, resulting in a decrease in lipoprotein clearance, and therefore resulting in an increase in abundance of cholesterol and plaque in the arteries. So we have our four experimental groups, 5 mice in each group. So our first group is our baseline group, which I'll explain more in the next slide, but keep in mind this group is 4 weeks younger to our next 3 groups. Our second group is our PBS group, or no treatment group, or no vaccine group. Our third group is our old vaccine, 008, which is the ApoB immunogen plus the dexamethasone adjuvant only. And our new vaccine, 3208, is the same thing plus the two new proprietary adjuvants. So our experimental design. So these mice are given birth and let to live for 12 weeks to come of age. And they're put on a high-fat diet, 40% fat for about 4 weeks. And group 1 is euthanized right after this 4 weeks, which serves as our baseline group because it is 4 weeks younger. So now groups 2 through 4 are put on a normal fat diet, which is about 4% fat compared to the 40% fat, and given their respective treatments. So there are 3 doses of the vaccine. So you give one dose every other 3 days, and it is an intramuscular injection into the rear right leg. So then after these 4 weeks have elapsed, groups 2 through 4 are euthanized. And again, they are 4 weeks older than our baseline group. So aim 1 is to determine whether or not immunization leads to a reduction of atherosclerosis as assessed by end-phase analysis in the aortic arch, which shows you the area of the lesion. So you give your mice their respective treatments, and you want to actually perfuse the blood with a type of saline called PBS. The purpose of doing so is to clear out all the blood from the body and replace it with a clear solution to make the lesion visible in the aorta. So next, you want to remove all the fat from this cavity here, and you can clearly see that perfusions actually work because the tube is a clear white color, and the lesion is the milky white color up top here. So now to perform end phase, you have to isolate the heart isolate the aorta from the heart. So you cut down in this cavity right here, cut the 3 arterioles up top, and cut it away from the heart. So now you can perform end phase by cutting open the aorta. And once you do that, you'll actually get this picture right here, and you can clearly see the lesion as the milky white substance. And now you're able to quantify now. So here we have 4 representative images of our groups. You can see the different amount of lesions in all the groups. And looking at our statistics, our old vaccine, 008 compared to our no treatment group or PBS, are not statistically significant at all, meaning there's not much difference in these two groups. But when looking at our new vaccine, 3208, there is statistical significance compared to 008 in PBS. And this shows that the amount of lesions has been significantly reduced with this new vaccine. However, comparing our 3208 group to our baseline group, over the course of 4 weeks, the amount of lesion only increased minimally, showing that 3208 actually suppresses or slows down the development of lesion in the aortic arch. So now our second aim is to determine whether or not immunization leads to the reduction of atherosclerosis as assessed by morphometric analysis in the aortic root. And this shows you the thickness of the lesion. So you start with your isolated heart, and you want to locate the 2 atriums right here and draw a line. From there, you want to go 1 millimeter below, and this is actually where you want to cut the heart to cut it with the blade. So now you'll get your top half of the heart, and you want to put it into a well. And now you want to get your OCT, or optimal cutting temperature medium, which freezes the heart at about -22 degrees. So you apply the OCT, and now you want to freeze it in the freezer for about an hour or so. After you freeze it, you want to take your frozen section of heart and take it to the cryostat machine, which sections your heart in very thin layers measured in micrometers. So now you can collect your sample on the slide. But actually, to visualize the lesion and the aortic root, you want to do some staining. So we first use oil red O, which stains lipids red, and this therefore will actually stain the lesion in a very dark red color. From there, you want to use HNE now, which stains the cell's nuclei blue. The purpose of doing this is to stain the purpose of this is to show that this is the real heart sample we're looking at. So after the 2 stains you complete, you'll get this picture under the microscope. You can clearly see this is the aortic root demonstrated by the tricuspid valve structure right here. You can clearly see the lesion has been stained in a very dark red color, and the cell nuclei has been stained in a purplish blue color as shown here. So again, you can see a similar trend as compared to the aortic root results. So you have 4 representative images of all the groups. You can see all the differences in each one. And again, shows that 3208 follows a similar trend in reducing the amount of lesion when compared to our old vaccine, 008, and our no treatment group. But when compared to our baseline group, over the course of 4 weeks, the amount of lesion only minimally increased, again showing that 3208 suppresses or slows down the development of lesion in the aortic arch. So we can conclude that our 3208 SI is shown that it's able to slow down the development of lesion of atherosclerosis greatly when compared to the groups that received no treatment and our old vaccine with only the dexamethasone adjuvant. And this shows that our new vaccine is actually very effective. So some future studies we'd like to conduct. So increase the time of high-fat diet to allow for more substantial development of lesion prior to the given vaccine. Test the mice blood plasma for cytokines to show the pro-inflammatory cell levels. And quantify the amount of antigen-specific T regulatory cells before and after to see if the vaccine actually raised T regulatory cell levels before and after or not. And finally, translate our research into therapy for humans in the near future. I'd like to take a moment to acknowledge my mentors, Dr. Chen and Dr. Gojing, for the opportunity they provided me, my lab partner, Jason Kaler, because without him, these end-phase results would never be possible, Dr. Ramaswamy, Janet, Jody, and most importantly, all the interns that made my experience really fun here, and I really appreciate all of you guys, and the College of Medicine and Thermo Fisher Scientific. Thank you.
Thank you, Varun. Our next presenter will be Maureen McCoy. She's an undergrad student. When she returns to college in this fall, she'll be a senior at University of Illinois at Urbana-Champaign. She's majoring in biochemistry, and her research and her career goals are to become a research scientist in an industry setting. She enjoys hiking, community band, gardening, and British comedy shows. Over the summer, she worked with Amarjit Flora, and Amarjit describes her as a simply awesome person. Maureen?
And now? Okay. Well, in case you didn't hear me the first time, I want to say how grateful I am for the opportunity to do research this summer and to be able to present our research today. And so the project we worked on this summer was automation of the phospho enrichment using magnetic iron NTA beads in the Kingfisher Apex magnetic particle processor. There we go. Thank you so much. All right, so since our protocol is for phospho enrichment, let's first talk about what phosphorylation is and why it's important. So phosphorylation is the post-translational addition of a phosphate group to a protein. And so these phosphate groups are added by kinases and removed by phosphatases. And this makes phosphate groups really important in cell signaling pathways where they act as a sort of molecular switch. And therefore, phosphate is also important in many diseases, especially cancer, autoimmune diseases, and also, as we'll see, neurodegenerative diseases. And so the tools that are typically used for studying phosphorylation, such as Western blotting and flow cytometry, are limited by the available antibodies. So this means that for each new type of phosphorylation that you want to study, you have to have an antibody specific for that. And so mass spectrometry, where we break our proteins down into peptides and study the ion fragments of these peptides, is uniquely suited for overcoming this challenge. And so with mass spectrometry, we can study thousands of proteins from one sample, and this is independent of antibody availability, and also we can identify novel phosphorylation sites. Okay. From the beginning? Yeah. So can we hear with this? Yeah. Okay. Hello? How about now? Okay. Is this good? Keep trying, keep trying, keep trying. Testing, testing, testing. Are we good? Okay. How far back would you? Just start a little bit here. Will it? Yeah, definitely. Okay. So, our project this summer was automation of phospho enrichment using the magnetic iron NTA beads in the Kingfisher Apex magnetic particle processor. So, protein phosphorylation is the post-translational addition of a phosphate group to a protein by kinases, and it can be removed by phosphatases. And so this makes phosphorylation really important in cell signaling pathways, where it can act as a molecular switch to turn a protein on or off. And this makes phosphorylation implicated in many diseases, especially cancer, but also autoimmune diseases and neurodegenerative diseases. And so typical tools for studying phosphorylation, such as Western blotting and flow cytometry, are dependent on antibodies. So if you want to study a certain type of phosphorylation, you need an antibody specific for that. And so with mass spectrometry, we can study thousands of proteins from one sample independent of antibody availability, and also we can identify novel phosphorylation sites. However, there is a challenge of studying phosphorylation with mass spec, and this is that phosphopeptides are low stoichiometry, so they're low levels of phosphopeptides compared to all the other peptides running through the mass spec, and they also ionize poorly. So in order to study phosphorylation, we want to do enrichment for phosphopeptides first. And so the tool that we focused on was IMAC, or immobilized metal affinity chromatography, using iron NTA. And instead of using iron NTA in a resin form, we used iron NTA chelated to magnetic agarose beads. So the advantage of using these beads, as Anna explained, is that you can use them with automation. And so we optimized the protocol using the iron NTA beads and the Kingfisher Apex magnetic particle processor. So we had three main objectives. The first one was evaluate the plastic leachables that come off of our existing Kingfisher plastics. The second was to optimize the actual parameters of the protocol. And finally, we wanted to apply this optimized protocol to cerebrospinal fluid samples. So this is our overall workflow. We start off with HeLa S3 nicotazole-treated cells, and then we process these with EasyPep chemistry along with phosphatase inhibitors in order to get our clean peptides. Then we enrich these peptides using the Kingfisher to get our phosphopeptides, which we inject onto the mass spec and analyze with Proteome Discover software. So we want to focus on the part that we actually looked at, so this is the Kingfisher phospho enrichment. So again, the important thing to remember with the kingfisher is that our peptides of interest are sticking with the beads, and these beads are moving up and down while the sample plates rotate up and down underneath. So the first thing we do is we incubate our beads with our peptide digest. We'll mix, and you'll notice when we lift our beads, our peptides will come with. So then when the wash plate rotates underneath, we can release our peptides into the wash. And you'll notice when we pick up our beads, then we now have our phosphopeptides. And then lastly, we release our peptides into the elution, which will cut the connection between the phosphopeptides and the beads. And when we pick up our beads, we're left with our clean phosphopeptides. So the first challenge that we came across is that the kingfisher was originally designed for DNA and RNA processing. So this means it's not completely compatible with the solvents that we're using for proteomics. And so immediately we noticed our samples were very difficult to dry, they were gelatinous, and they were colored. And so this is an issue because this means that plastic contaminants are coming into our samples, and they'll interfere with our signal, suppress our signal, and we won't see as many peptides in the mass spec. So in order to determine what leachables we had, we first ran a blank, where you can see there are a couple of contaminant peaks, but it's nothing unacceptable. But then we ran our wash buffer, which was incubated on our kingfisher deep well plates, and you can see there are numerous plasticizers and detergents which will interfere with our mass spec data. So in order to resolve this problem, we first tried doing an extra desalting cleanup after our enrichment. And so here on the left here, you can see average phosphopeptide IDs. So in the first lane here, we have our manual, which is a similar protocol, except run on a magnetic stand instead of on the kingfisher. And so you can see we have about 3,500 phosphopeptide IDs. However, when we do cleanup, the number of phosphopeptide IDs actually increase. And you can see compared to cleanup, the number of phosphopeptide IDs increases almost fourfold. So this cleanup is really important to making sure we aren't suppressing our signal. And additionally, we have phosphopeptide specificity. So this is the ratio of phosphopeptides to non-phosphopeptides, or to the total phosphopeptide pool. And you can see that the specificity is greater than 95%. However, the trouble with cleanup is introducing a cleanup after enrichment causes a lot of variability, and it reduces our yield. So we also wanted to try rinsing the elution plates prior to enrichment. So in this picture here, you can even see with including the rinse, our samples just look much better. They dried faster and they look a lot cleaner. And additionally, you can see we had improved phosphopeptide IDs with the rinse and high phosphopeptide specificity throughout. So the bead ratio is the first real parameter that we looked at in our kingfisher optimization. So we tried 3 different ratios, 1 to 10, 1 to 20, and 1 to 50. And you can see that phosphopeptide IDs are pretty consistent throughout the manual and our 3 different ratio samples. However, when you look at phosphopeptide specificity, our lowest bead ratio, this 1 to 50, actually had the highest phosphopeptide specificity. So this is the ratio that we chose to continue with. And then next we compared the wash volumes. This is for the organic washes. So we looked at 500 microliters and 100 microliters. And you can see that there's a slight increase in IDs and specificity for 100 microliters. So this is what we chose to continue with. And then finally, we wanted to apply our optimized protocol to our CSF samples. So we had samples from 5 different patients, 2 normal, 1 Alzheimer's disease, 1 with mild cognitive impairment, and 1 with Parkinson's disease. And we did these in triplicate along with a pool. So we had 16 samples total. So then we processed our samples again with EasyPep chemistry and phosphatase inhibitors to get our peptides. But before we did our cleanup, we labeled with TMT Pro 16-plex. So this allowed us to pool all of our samples together and before mass spec. And this also allowed us to do relative quantitation between our different samples. So some samples we enriched using our optimized protocol, and other samples we left unenriched. And here you can see that with enrichment, we were able to see 202 more phosphopeptides, and our specificity was able to increase to 54%. So here that you can see that enrichment is essential if you want to study phosphopeptides from these samples. And here we were able to look at the relative quantitation between the different samples. So here in our PCA plot, you can see that each sample formed its own distinct population, indicating the reproducibility. And then we were also able to find an interesting protein that was only phosphorylated in our enriched sample, and this was fibulin 1. So fibulin 1, as you can see, is decreased abundance in our disease samples. So we have Alzheimer's, MCI, and Parkinson's. And this is really interesting because fibulin 1 has been found to bind to amyloid precursor protein. And as you may know, amyloid precursor protein is implicated in these neurodegenerative diseases. So finally, in summary, we determined that kingfisher contaminants can be removed either with cleanup or preferably with a rinse. And we also optimized our protocol parameters and applied this optimized protocol to our CSF samples to identify more phosphopeptides and differentially express proteins such as fibrillin-1. And so ultimately, we hope that this protocol can be used by researchers to study phosphorylation in high throughput with little variability. I'd like to thank my mentor, Amarjeet Flora, even though she can't be here today. She put in a lot of time and effort into this internship. Also, special thanks to Bhavin Patel for all his patience and guidance and everyone else on this list. Thank you so much to all the R&D staff for making this a really great summer and being so kind and welcoming. And I'm happy to take any questions.
So this is very interesting project because the phosphoprotein peptide has some very interesting roles in biology and also disease. So my question is before you label this using the bead to label this phosphopeptide, you also lysis and also digest.
Mm-hmm.
So my question is because do you need to digest to certain length? Or to digest, is there optimal the length for this peptide for you to be more efficiently to process?
So digestion time, I guess, depends on how much sample you're running. Usually we do between 1 and 3 hours, but we do want pretty much complete digestion. So we usually look for greater than 95% zero miscleavages. And
Okay.
so it's really important that they're broken down into the peptides so we can analyze them properly with the software.
Okay, okay.
So in one of your slides, you showed that you had a choice between titanium dioxide and iron, and you chose to do iron. Was there a specific reason for choosing iron, or was that what was available?
It was more of a situation of what's available. Titanium oxide and iron NTA do produce slightly different populations of phosphopeptides, and there are some protocols which use sequential versions of either of those, but for our experiment, we just focused on iron NTA because of availability or what products need to come out.
Nice talk. Did you happen to compare TMT-labeled peptides versus unlabeled peptides in the purifications or in the enrichment strategy?
So we did not specifically compare. The only TMT labeling we did was with that final application. That would be a future step. Also, we'd want to test with fractionation also, especially on those CSF samples. But yeah, that would be a future direction, but not something we did this summer.
The point about the plastic leaching into the sample is very interesting. So does this happen by long-term storage of the sample in the plastic containers? And after you alluded, is there any special type of plastic containers we should use so that we don't have leakage of these products into the sample?
So for what we used, they weren't particularly on the plates for long term, but we would still see signal suppression. I know some people will freeze their samples down on the plate and leave them like that, and I presume that the plastic leaching would only get worse if you do that. But normally we speed vac our samples down, and so the leaching would be less of an issue after that point. We are looking into getting new, better plastics for the kingfisher that are compatible with proteomics, and one option for those are plasma-treated plastics, but that's still something to come in the future.
Thank you.
Our next high school speaker is Dhruv. Dhruv will be a senior at Auburn High School in fall. He intends to pursue a career in biomedical sciences. For hobbies and interests, he enjoys playing tennis and basketball, and I've heard that he likes to read as well. Over the summer, he worked with Dr. Neelu Puri, and Dr. Puri describes Dhruv as inquisitive, agog, and a budding scientist. Dhruv?
Hello? Oh, there we go. All right. Thank you, Dr. Janaki, for the intro. Hi, everyone. My name is Dhruv Ramaswamy. I had the privilege of working in Dr. Neelu Puri's lab this summer, and we studied the role of sphingosine kinase 2 in TKI resistance and tumor progression in non-small cell lung cancer. So lung cancer is a leading cause of all cancer-related deaths, with over 200,000 cases and 100,000 deaths projected in 2021 alone. Now, the most prominent form of lung cancer is non-small cell lung cancer, which accounts for about 84% of all lung cancer cases. So in early stages, non-small cell lung cancer is usually treated with surgery and then tyrosine kinase inhibitors, such as erlotinib and opaganib. And what these tyrosine kinase inhibitors do is that they inhibit the tyrosine kinase domain of EGFR, which is a growth factor receptor that stimulates cancer cell proliferation, and inhibition of this induces cancer cell apoptosis. So the issue comes when, after 6 to 18 months, patients eventually develop resistance to the drug. And reasons for this resistance are quite unknown, but they are attributed to EGFR mutations, new TK domains in EGFR, and a T790M mutation. So due to the issue of TKI resistance, many EMT biomarkers and proteins are being studied to eventually develop molecular targeted therapies against TKI resistance. And we specifically studied sphingosine kinase 2 because its properties in TKI resistance are vastly unknown. So a little bit of background on sphingosine kinase 2. Sphingosine kinase 2 phosphorylates sphingosine into sphingosine 1-phosphate, and sphingosine 1-phosphate is associated with PHB2 in mitochondria, which regulates mitochondrial respiration and apoptosis, ERK1 and 2 in the cytosol, which is activated to the MAP kinase pathway when several membrane receptors are activated, and it inhibits HDAC1 and 2, which regulates general gene expression in the nucleus. So we hypothesized that sphingosine kinase 2 may be involved in inducing TKI resistance in non-small cell lung cancer cells and may be correlated with tumor progression, and inhibition of sphingosine kinase 2 can actually overcome TKI resistance. So to do this, we first investigated the role of sphingosine kinase 2 in TKI, erlotinib and osimertinib resistance by comparing it to drug-sensitive cells, which are known as parental cells. So what we did first is that we treated the parental cells with specific drug concentrations for both ER and OR, and we kept them in specific cell lines for experimentation. This includes H358 and H2170, specifically for ours. And we first wanted to see the gene expression of sphingosine kinase 2 in TKI resistance. And we did qPCR to do this. So first, we simply plated the cells in a 60-millimeter dish. Next, we let the cells grow for a little bit. And they're giving starving media after reaching a desired confluency of 90%. Next, it was incubated for 24 hours, and we went through the mRNA extraction process where triazole and chloroform and everything was added. And this mRNA was converted to cDNA, which is then used in the qPCR setup with the CyberGreen master mix. And then we ran it in the qPCR for 40 cycles, eventually giving us a specific CT value that we used to calculate the fold change. So the qPCR results indicated that there was clear upregulation in OR and ER cells for both cell lines H358 and H2170 For cell line H358, there was a 1.3 and 1.16 increase in ER and OR cells respectively. And for H2170, there was a 1.3 and 1.5 increase in fold change for ER and OR respectively. So we use the primer GAPDH as a housekeeping gene in this scenario, and the p-value is significantly under 0.05%, indicating statistical significance. So this concludes that sphingosine kinase 2 may be involved in TKI resistance. But we also wanted to see the protein expression of sphingosine kinase 2 in TKI resistant cells. So we did this by first collecting lysates for the experiment for Western blotting, and we then did gel electrophoresis, and we transferred it to a membrane and blocked with BSA to prevent against any nonspecific binding. Then we simply added the primary antibody so it would bind to the target protein, and we added the secondary antibody, which had HRP, which then interacts with the ECL substrate. It's an oxidizing process, which then gives us a sphingosine kinase protein expression that we're looking for. So the results of the Western blot indicated that there was a 1.5 and 1.46 increase in fold change for OR cells for cell lines H358 and H2170 respectively. In this case, beta-actin was seen as the loading control, and this process was regulated through the addition of EGF and osimertinib in different places. So this indicates that the sphingosine kinase expression levels are upregulated in these OR-resistant cells. Additionally, we performed an immunofluorescence test on cell lines H358P, OR, and ER, and we're able to see clear upregulation in OR and ER cells by about 1,500 fluorescent units, or twofold. And we can even see this as well, visually as well. The FITC is simply the sphingosine kinase expression. We see a nucleus stain of DAPI, and then we're able to see an overlay here. So now that we know that sphingosine kinase 2 is associated with TKI resistance, we now can test the efficacy of a sphingosine kinase 2 inhibitor on cell viability of cancer cells to observe the potential of a sphingosine kinase inhibitor to overcome this resistance. So what we did here was an MTT assay where we had a control well which had no cell death, then a drug well, osimertinib, then a sphingosine kinase inhibitor well. This was all in a 96 well plate, and the sphingosine kinase inhibitor well is opaganib, and a drug plus inhibitor wells were also used to test the additive effects. And this was all analyzed in a spectrophotometer at a wavelength of 570 nanometers. So results of the MTT indicated that there was an increase in inhibition for all the different types of wells. This included drug, inhibitor, and drug plus inhibitor. And specifically, there was a 71% inhibition for OR cells for drug plus inhibitor, and for parental, there was a 70%. But more importantly, the drug plus inhibitor for the resistant cells was greater than the drug and inhibitor individually by about 9%. So this indicates that the sphingosine kinase inhibitor actually enhanced the performance of the osimertinib drug overall on cancer cell death, thus showing sphingosine kinase inhibitor to be a promising suppressant of TKI resistance. Finally, we wanted to go from simply cancer cells to now specific cancer tumors to add a little extra layer of complexity. So to do this, we studied the correlation of sphingosine kinase 2 expression in tumor progression by comparing early and late stage patients. So we did this through the IHC process, where we simply obtained the patient slide first. We dehydrated it with multiple ethanol washes, and we retrieved the antigen. We then did a protein block to prevent any nonspecific binding. And we simply added the sphingosine kinase primary antibody, which was made in rabbit. Binded, and then we add the secondary antibody, which is actually biotinylated, and because it was biotinylated, we simply add peroxidase with streptoavidinin, and the HRP is bound to the streptoavidinin, so the HRP reacts with DAB substrate to give us a brown sphingosine kinase 2 expression. So we can see this expression in varying types of patients. So first, we use a control with normal skin, normal lung, normal thyroid, and a tumor-negative control, which doesn't include primary antibody. We see that there is basically no sphingosine kinase expression anywhere to be found. There's no brown anywhere, except for the nucleus stains of blue. Then in early stage, we see more sphingosine kinase expression. And then for late stage, we see an increased amount and the most amount of sphingosine kinase expression. And one may also notice that sphingosine kinase 2 is localized in the only the cytoplasm in early stage, but it's all because it only goes around the blue nucleus stains. but in late stage, it's localized in the nucleus and the cytoplasm. Additionally, we were able to quantify our results as well by looking at the average brightness levels of 13 early and 13 late stage patients. We're able to see that the average brightness of early stage was much greater than the average brightness of the late stage patients. And because the greater the average brightness, the less sphingosine kinase expression because the less dense brown, thus indicating that sphingosine kinase 2 does play a role, is upregulated in tumor progression and is tumorigenic. These are simply the demographics for the tumor samples we use. So in conclusion, sphingosine kinase gene and protein expression is upregulated in ER and OR cells as studied by qPCR, Western blot, and immunofluorescence, thus indicating its correlation to TKI resistance. The inhibition due to the combination of the drug osimertinib and sphingosine kinase inhibitor opaganib was more than additive compared to the drug inhibitor individually, thus indicating that the sphingosine kinase inhibitor enhanced the drug's performance. And finally, higher expression of sphingosine kinase was observed in late stage than early stage. Thus, we see that it can be used as a good prognostic biomarker for IHC. So future directions include studying more sphingosine kinase in signaling proteins such as ERK and HDAC. We can also study the correlation of EMT processes to see if sphingosine kinase itself is an EMT blood biomarker. We can also study the role of sphingosine kinase in smokers versus non-smokers through further IHC staining. And we can determine sphingosine kinase 2 expression in tumors and cells with EGFR TK mutations. Finally, I just want to thank Dr. Puri, who is my mentor, Dr. Ramaswamy, Jody, Thermo Fisher Scientific, UIC, College of Medicine, Namrata Dubey, and Katie Lam, as well as the rest of the MBT students and my lab partner, Caleb Rathman. Thank you.
So we'll take a 10-minute break at this time. So it's around 3:05. So let's meet back at 3:15. There will be cookies and I think brownies outside. So please help yourself. Thank you. welcome back. Hello, everyone. Hello, everyone. I think it's time to get started. So there was just one announcement to make. No food is allowed in the auditorium. I'm sorry. Yeah, so please don't eat inside the auditorium. Feel free to step out. Sorry about that, and thank you. So let's start the second half of the program. Our presenter right now would be Puranjay Gupta. Puranjay will be a senior at Auburn High School this fall. He's interested in pursuing a career in biomedicine. In his free time, he enjoys playing golf and guitar. Over the summer, he worked in Dr. Matthews' lab, and Dr. Matthews says that his intern is a self-motivated scientist. Puranjay.
Hello, can everyone hear me? All right. Thank you, Dr. Janaki, for the wonderful introduction. Once again, my name is Puranjay Gupta, and I had the privilege to work under the guidance of Dr. Matthew this summer about cell response and survivability of implants under inflammatory and infectious chemical environment. Successful joint replacement is a life-enhancing procedure. As a result, the incidence of joint implantation is profound, with nearly 1 million combined total hip and knee replacements. Additionally, joint implants can be used in a variety of other fields, such as dentistry, with some sources finding nearly 5 million dental implant procedures each year. However, with the growing usage of implants also comes a growing number of implant failures. In fact, recent data has found the leading cause for implant failure as infection and inflammatory reaction, accounting for nearly 20% of all implant failures. So let's take a closer look at what implant infections really are. There are 4 main components to consider when evaluating implant infections. The first major component is inflammation. Some studies have found that metal hypersensitivity can occur, where local adjacent tissue can react negatively with metal prostheses and result in inflammation. Additionally, these studies have noted that you can simulate this inflammation through hydrogen peroxide, a substance known to induce oxidative stress and cause inflammation. The next major component of implant infection is the infection part itself. Recent studies have found that small pathogenic bacteria can form complex communities of microorganisms on the surface of implants called biofilms. As a result, these biofilms can both protect these pathogenic bacteria from antibiotic treatment, as well as create highly intensive lactic acid environments, a natural byproduct of the cell's metabolism. The next major component of infection stems from the body's own immune response to infection. In fact, a study here from the own UIC found that the macrophage secretion could potentially accelerate the corrosion of these implants. Additionally, the last major component to consider is tribal corrosion, which is a physical and chemical process that results in the degradation of metal ions and metal particles that can cause and increase the severity of implant infection. Therefore, we've seen that joint infection continues to cause an increase in implant failures, and that there's a lack of existing research regarding the interactions between cells and implants in an infectious environment. Therefore, in our study, we hypothesize that implant infection and inflammatory conditions will affect the viability of osteoblast cells, and that macrophages challenged by these conditions will alter the corrosion rate of titanium implants. In order to test our hypothesis, we'll split our experiment into two major aims. Within the first aim, we'll evaluate the effect of various ion concentrations on the viability of cells before evaluating this viability under conditions of inflammatory and infectious compounds. And for our second aim, we'll analyze a change in implant corrosion when exposed to compounds that simulate the body's own immune response to infection. In essence, we'll examine the impact of implant infection on cells and the impact of implant infections on the titanium samples themselves. But before we begin our experiment, we must first consider our experimental conditions. A recent study conducted here at the UIC found that there were extremely low pH conditions reported in implant infectious environments. As a result, we managed to emulate these conditions through a concentration of 0.23 microliters per milliliter of lactic acid. This same study also found presence of a high number of local oxygen reactive metabolites, a situation we were similarly able to emulate through a concentration of 0.5 millimolars of hydrogen peroxide. The last major component to consider in our study is the presence of metal ions. However, due to the recency of our own research, there's a limited amount of information regarding the presence of metal ions in implant infection. Therefore, we must conduct our own experimental analysis to determine what titanium ion concentrations most closely match that of implant infection. This takes us to the first part of our experiment. In this experiment, we'll culture MG63 osteoblast cells before exposing them to a variety of titanium ion concentrations, and then evaluating cell viability through an Almar blue assay. Our results are as follows. You can see a general steady decline from concentrations from 0 to 20 parts per million titanium ion concentration, as well as a more significant drop between 20 and 50 parts per million. Therefore, we choose to use titanium ion concentrations of 20 and 50 parts per million in order to emulate an implant infectious and inflammatory environment. For our emulation experiment, we'll both culture MG63 osteoblast cells, as seen before, and then treat them with the aforementioned concentrations of hydrogen peroxide, lactic acid, and a combination of both solutions, repeating for the previously determined titanium ion concentrations. Additionally, we'll judge cell viability through the Almar blue assay, as well as live dead image staining. Our cell viability results are as follows. In general, we noted that hydrogen peroxide tended to be more toxic to cells as opposed to its lactic acid counterpart. Additionally, we also noted that the combination of both solutions tended to be the most toxic to these cells. We did notice some interesting features. For example, the hydrogen peroxide actually decreased in toxicity at 20 parts per million titanium ions, as opposed to 0 parts per million. If we look at our live dead image staining, we can affirm these results. For instance, if we take a look at the 0.5 millimolar hydrogen peroxide at 20 parts per million titanium ion, we can see that relative to the control, it's a slightly darker shade of green, indicating less viable cells. If we progress downwards to the 50 parts per million, this shade of green turns even darker, indicating yet even less cell viability. If we move on to the right, we can see this trend similarly shown throughout the lactic acid, and even more pronounced throughout the combination of both solutions. Now, if we take a step back, we can see the drastic cell disparity between the control as well as the conditions shown in figure G. Therefore, in our part 2 of our experiment, we hope to expose macrophage cells to the conditions in part G to emulate the body's immune response to infection. Within this corrosion testing, we'll first grind and polish our titanium ion samples as shown above, and alongside this, culture our raw macrophage cells in a control medium, as well as the highly infectious compounds. Then we'll run our corrosion testing in a corrosion cell and analyze our data through a potentiostat. The first set of data that we've gathered comes from the potentiodynamic test, and from this test, it conveys data known as cyclic polarization. From the cyclic polarization, we're able to derive our corrosion potential, or E core, and our corrosion current, or I core. As we can see in the data, both the challenge has a higher value in the E core and the I core, indicating that it underwent higher corrosion. The second set of data that we've gathered comes from electrochemical impedance spectroscopy. And from this, we can derive our Nyquist and Bode plots. And from this Nyquist and Bode plots, we're able to gather both our resistance and capacitance values. We can see that the control has a higher value in each of these categories, which provides further evidence of increased corrosion on the challenge surface. The last set of data that we collected were scanning electron microscope images. In the figure shown above, you can see that there are slight remnants of a titanium oxide layer, which we speculate means that the challenge surface underwent an increased rate of corrosion. Additionally, on the challenge surface, you can see these small white dots, signifying possible holes within the surface, evidence of pitting corrosion. In order to discuss our results, we can look at possible reactions that occur between these compounds. In our aim 1, we believe that there are possible reactions that result in the increased toxicity of several compounds, resulting in some of the observed toxicity. We also noted neutralization reactions, which could possibly explain some of the observed neutralization phenomena. Additionally, if we move on to our second aim in the corrosion testing, we speculate that our challenge media formed a sediment layer on the surface of the titanium implant, indicating a higher corrosion potential, but ultimately causing a higher rate of corrosion. In order to better understand our results, let's take a look at a clinical implication. This example that I've provided is called peri-implantitis, which is an example of a dental implant infection. As we can see, titanium implants can corrode, aggravating some gingival cells. In turn, this can create a highly infectious environment that incurs secretion of macrophages. In result, these macrophages can accelerate the corrosion of titanium implants, leading to a cyclical effect of accelerating corrosion and cell toxicity. In conclusion, these results have validated our hypothesis. In our aim 1, we saw a decrease in implant in cellular survivability due to implant infectious and inflammatory conditions. And in our aim 2, we saw an increase in titanium corrosion due to the body's immune response. Overall, more research must be conducted to further verify our findings, as well as minimize implant infection. I'd like to take this time to thank my mentor, Dr. Matthew and Dr. Kai, as well as my co-mentor, Dr. Vijay Kumar, as well as the other people on the screen. I'd also like to thank our sponsors, the Blazer Foundation, the NIH, as well as the Thermo Fisher and UIC. Thank you for your time.
Thank you, Jay. Now we're going to hear from Scott. He's our final undergrad presenter for the day. Scott will be a junior at Johns Hopkins University this fall. He is double majoring in molecular and cellular biology and computer science. His career goals are to pursue a career in computational biology or bioinformatics. His interests and hobbies are riding his bike, listening to music, and learning new cooking recipes. Over the summer, he has worked with Dr. Paul Haney and Dr. Surbhi Desai, and his mentors say that he is an awesome scientist. Scott?
Is this working? Okay. Thank you, Dr. Janicki, for the wonderful introduction. I would just like to start by introducing my project that I worked on this summer called an assessment of surface capture methods for separated protein immunodetection for the purpose of improving Western blotting. So an introduction to Western blotting, it's one of the most popular protein detection assays since it was discovered in 1979. Mainly because of its separated protein, the ability to use separation of proteins. And around 5 to 8 million blots are performed each year. However, it's broken into 3 steps: the transfer step, the separation step, the transfer step, and the detection step. And these 3 steps add together to make a largely hands-on and time-consuming workflow that makes it hard for someone to multiplex. For example, if you have an experiment with 8 samples and 12 targets, you can see how quickly the membranes will add up and how messy your workflow will get. An estimation of this time will be 30 to 40 hours, and about 10 to 15 of those hours will be you with your hands-on working on the experiment. And this large hands-on time gives a lot of room for someone to make a mistake in the experiment. And if a mistake is made, the experiment will have to be reproduced, and this makes it difficult. Additionally, your results are only qualitative. You only get a darkness of your band rather than a number for your protein expression. However, one of the most variable things for a Western blot is the transfer step, where the proteins are transferred from the gel to the membrane. And this is inconsistent because proteins of low molecular weight will commonly blow through the membrane, and proteins of higher molecular weight will sometimes not transfer from the gel, will not come off the gel. Other times, proteins will fall off the membrane as well. And due to a poor transfer, your results will also be negatively affected. So making sure that you improve this transfer step early on will allow you to improve your results. So what the Rockford thermostat is looking for is to improve the Western process by developing a new technology that maintains size separation, but introduces quantitative protein detection on a surface that's not a membrane. So what we first started to do was separate our crude lysate by molecular weight, and then immobilize our proteins on a surface that's not a membrane, and then finally probe for a specific target. However, I cannot go more into about the separation due to confidential issues, but this is the general workflow of what we were looking for. So instead of membrane for a capture surface, we looked at fluorescent magnetic beads and microtiter plates. And the means by which to immobilize our proteins onto the surfaces includes a metal chelate surface, which binds proteins through hydrophobic and ionic interactions, a nutravidin biotin complex, which will bind biotin strongly, and an antibody, which recognizes antigens present on a protein. So for our plate, we had two options: a nutravidin-coated plate, where each well of the plate is coated with nutravidin, which can immobilize your biotin-related protein, and then from there you can come in with your primary and secondary detection to probe. For our other option, we had an antibody-coated plate, where the wells are all coated with a secondary antibody, which will capture your primary antibody. And after that, your biotin-related protein will be added, and a streptavidin HRP will be used to detect. So for these two options, we looked at which one would be a better fit for us. And from these graphs, you can see that the signal of the antibody-coated plate is much higher than the nutravidin-coated plate. Additionally, the background is lower, and the sensitivity is higher on the antibody-coated plate for all four of our targets that we looked at. We looked at cyclophilin B, GAPDH, HSP90, and ERK12. So from this, we concluded that we should move forward with the antibody-coated plate for our studies. So one thing we wanted to look for was to use targets of a wide molecular weight range, because we know that a Western may potentially struggle with lower end or higher end molecular weight proteins. So on the lower end, for P23 and GAPDH, you can see that we were able to detect both of these targets in the samples we expect, and we're also able to verify this through a Western blot. Additionally, for our higher weight molecular proteins, Q80 and vinculin, we can see the same thing on our plate, being able to detect them in the samples that we were looking for. And again, we were able to verify this with a Western blot. So we can say that targets of a wide molecular weight range are able to be detected with our plate mechanism. On beads, we have two choices as well: a nutravidin bead and a metal chelate-coated bead. The difference being the nutravidin bead will be coated with nutravidin, which will bind a biotin-related protein, whereas a metal chelate-coated bead will directly bind our lysate protein without need of biotinylation. So if you look for our lower molecular weight targets, both P23 and GAPDH can be detected in the samples that we expect for both nutravidin bead and the metal chelate bead. For P23, it is around 11, 10, and GAPDH will be 17-ish. And for our higher molecular weight proteins, Q80 and vinculin, we see the same thing for our metal chelate-coated bead and nutravidin bead. So we can say that both the plate, the metal chelate, and the neutravin bead are able to detect proteins of a wide molecular weight range. So in conclusion, we see that both our capture surfaces, the plate and the bead, are capable of detecting our proteins much simpler and quantitatively compared to a traditional Western blot. And as a user, this provides you a lot of advantages and options for what you want to use for your protein detection. For example, if you like to use a bead, you can use the bead to multiplex if you're looking to survey a lot of protein targets. And this would be mainly towards a high-end academic or a biotech company. However, if you were looking for a low-cost alternative and only wanted to survey maybe a couple samples, a plate would be a good idea for you as well. Because on a plate, you would only need a plate reader to detect, and many labs currently already have a plate reader. And finally, if you do not want to biotinylate your protein, you may use a metal chelate to remove the need of biotinylation. So moving on with our project, we would like to use the metal chelate option on the plate. So currently we're just using it on a bead, but the option of a metal chelate surface on a plate would also be a good idea to look forward to. Additionally, we will only use 4 targets for our assays, and it would be nice to investigate more than 4 targets just to try a larger molecular weight range. And currently we're using Western validated antibodies for our assays, and it would be good to identify validated antibodies specific to our assays. And finally, to reduce the amount of times that a user would have to interact with their experiment, we would like to develop an automated solution to this, much like the Kingfisher for DNA purification. And I would like to thank everyone on the screen that I listed. They're all very helpful for my presentation and my project, and I wouldn't be able to have done all this work without their help. And I would very much like to thank Thermo Fisher and UIC for hosting this event.
Okay, so I'm going to start with a question. We got a question from one of our attendees, virtual attendees. I'm very glad. So this question is from Brian Webb, and he wants to, what he asks is, why do you think you see higher signal on streptavidin plates than metal chelate plates for some protein targets and lower signal for the other targets?
Okay. Okay, I guess we can go back. So, an explanation for why you may see a higher signal for certain targets on the neutravin bead, but a lower signal on the metal chelate bead may be due to the differences in the instruments that we use to measure these targets. So, for the neutravin beads and the metal chelate beads, we used a Luminex machine, the FlexMap 3D and MagPix. However, some of our other data have been taken from another Luminex instrument. So the differences in instrument may also account for the reason why some may be higher than the others.
Yes, so I think it's very interesting to use this non-membrane to test the Western, test the protein expression. So my question is for some conditions, you may have 2 isoforms, for example, 2 different sized, which you can see by developing the membrane. My question is in your using this kit the condition, do you think this could be potentially in the future to distinguish these 2 different isoforms, or you will need 2 specific antibodies for this application?
So because our samples are separated by molecular weight, I think that we should be able to detect multiple isoforms in our graphs. Just there would be more than one peak in our graph. Maybe it would be later on or before the current peak. So this should be able to detect multiple isoforms.
Okay, I really liked your presentation, Scott. I think you have a very good project because I think we all in the labs, we constantly having this problem of the transfer, transferring the small proteins and not transferring the large ones. This is a constant problem. I had a question, so the beads you're using, have you quantified the results of the beads with what you get on a Western? Like, have you compared the two to find out whether they are similar or they are different? And the second question I have is that how much of, I know you're trying to do this automated system, which I think is a great idea because automation brings out the handler error. But then till it's automated, do you think that, because I know I work with magnetic beads from Thermo Fisher all the time, and there's handling error with beads. Some of the beads get stuck on the walls here and there and this and that. So how much of handling error do you think you have in your technology?
So I'm going to have you repeat the first question because I didn't hear it fully, but the second question you were asking about handling error.
Right.
So I believe that each time that you do interact with your experiment there, that it does increase the chance of handling error. However, on a Western, I believe there is just more room for error due to the inconsistencies of transfers. So I think with our beads and plate, there is less handling error, but yeah, the reason why we are introducing automation is to reduce the handling error. So yeah, I believe that our systems right now are not completely foolproof, but it is an improvement to Western blotting.
So the first question which I asked you was, did you compare the quantification of the protein which you got from the beads with that which you would get from a Western blot?
Yes, so we did look at a Western blot to verify our results. So we ran the samples on a Western blot to see that, yes, they were present in the samples we expected, and also on our beads, we saw that they were present in the samples that we expected as well.
It's a very interesting effort. I had a couple of questions. So do you understand what the mechanism for the metal capture?
Yeah.
How the chelator is actually capturing the proteins?
Yeah, so if you have a surface, for example, a bead, the surface will have chromium ions on the surface, or the bead will have chromium ions on the surface. And when you activate it with a metal chelate surface, the metal will bind to the chromium ions, which will make it a positive surface charge. And for a protein will commonly have a negative surface charge, so the two will attract each other through just surface charge, I believe.
Another question, do you have an idea why, if you immobilize a secondary antibody to capture on a plate versus the Nutravidin, and I mean, you're using the same components, Nutravidin and a capture antibody. Any idea why the order matters, like Nutravidin on a surface versus the antibody on the surface for the capture of the target?
I believe for the Nutravidin, you will require the protein to be biotinylated, but also any free biotin must be removed because if there are free biotin present in your samples, they will cover up the Nutravidin binding spots. However, for an antibody-coated plate, the need to remove free biotin is reduced.
I see, and you can wash away the free biotin in the
Correct,
process.
correct.
Okay, very good. And then the other question is, when you run an SDS, page gel, you typically reduce and you denature with SDS, whereas for the biotinylation and this capture and the antibody approach, am I correct in assuming there's no denaturation? I assume not, or I assume you're not denaturing. So that comparison of results, it seems like it's going to be pretty different. A denatured protein separated on an SDS page is going to look quite different than in a native protein, more or less, on a bead. So anyways, I don't know, any comments on that? I'm not sure if that's a question. I apologize.
I do have an answer, however, I don't think I'll be able to go into it based on confidential reasons. My apologies.
We can talk to you once.
Yeah.
Oh, no, that's okay. I think there was one last question. Let's take it from Dr. Ramaswamy.
Very interesting, the technology. Short period of time advances the whole Western blotting protocol. So one of the questions I have is that since you get the native protein bound to the plate and then you're reading that, is it possible that for the downstream, can you pull out that protein out of the plate, for example, doing mass spec or any of those application?
Sorry,
because it is already purified, right?
Could
When
you
you
repeat
think
the
about
question?
that transferring to another membrane and then pulling out of there, if you could do that right, that would be wonderful.
Sorry, I couldn't hear your question. Could you repeat it again?
You didn't understand my question?
Yeah, I couldn't hear it. I'm sorry.
Oh, okay, I'm sorry. So you are actually binding the protein with the antibody onto the plate and detecting, right? So I'm asking downstream, can you do mass spec analysis or anything on that one? So that also substantially reduces further handling of the protein. So in Western blotting, once you have done, if you wanted to pick any protein and do a mass spec and subsequent analysis, can be potentially done through this technology as well.
That is a question that I'm not sure. I believe we could use mass spec if we wished. However, yeah, I think that we could, but that would, I'm not quite sure.
Okay.
Thank you, Scott. Our final presentation for the day will be from Shreya Vatam. Shreya worked in Dr. Divya Bijikumar's lab over the summer. She's going to be a senior at Hononegah High School when she returns to school. She's interested in pursuing a career in medicine or something to do with healthcare. In her free time, she enjoys dancing, cooking, spending time with her family, friends, and especially her cats. Shreya? Oh, her mentor says that Shreya is inquisitive and an avid learner. Her excitement for science was encouraging and supportive to tackle the challenges we encountered. Shreya.
Can you hear me? Oh. All right. Thank you for the introduction. I'm honored to close out this forum with presenting my research on the suppression of inflammatory responses using an alpha-fluorophane-loaded polylactic co-glycolic acid nanodrug delivery system for rheumatoid arthritis. To start off with some general background, RA is a chronic inflammatory autoimmune disease in which the body's immune system attacks its tissue, mainly targets the joints though. RA is not curable, but if not treated, it can lead to accumulating joint damage and irreversible disability. As seen on this map to the right, about 1% of the world's population has arthritis, and women are 2 to 3 times more likely to get RA than men due to the stimulatory effects of estrogen on the immune system. And in general, around 60% of all arthritis cases are due to genetics. Here we can see the pathogenic aspect of rheumatoid arthritis. First, an unknown trigger promotes inflammation in the blood vessel, attracting leukocytes into the tissue. The T cells then activate the macrophages, which results in the release of the pro-inflammatory cytokines. These cytokines then induce rank ligand and MMP due to the fibroblasts, and in result, rank ligand produce osteoblasts, and the MMP degrades cartilage, which results in joint degradation and destruction. So the current management for rheumatoid arthritis is through the use of disease-modifying anti-rheumatic drugs, or DMARDs. There are 2 types of these drugs: synthetics, which can only be taken orally, and biologics, which can only be taken through injection. Methotrexate, which is a synthetic, is the current gold standard disease-modifying anti-rheumatic drug, and only has a 20 to 40% efficiency rate by itself. But when combined with a biologic, like Etanercept, an extra 20% efficiency is added on top. However, these current strategies do have adverse side effects, whereas these drugs are toxic to the body. So, a safer, non-toxic drug is imperative for treatment for rheumatoid arthritis.
This is why we're choosing to use alpha-fluoropane as our drug of choice. Alpha-fluoropane is a natural compound with proven anti-inflammatory properties. You can naturally get this drug by ingesting cruciferous vegetables like broccoli, Brussels sprouts, and cabbage. But just ingesting these vegetables will not be enough to give you the anti-inflammatory properties as seen with this drug. This drug has also been widely researched regarding other diseases like cancer, diabetes, and allergic airway diseases, and can also be found through the NRF2 pathway as seen on the right here. There have also been possible anti-arthritis effects found with using this drug, as seen with a study done in 2018. However, a general problem is seen with the effective delivery like DMARDs like alpha-fluoropane, so an effective drug delivery system is desired. This is why we are choosing to use nanotechnology for drug delivery. Nanoparticles themselves range around 100 nanometers in size and can be found in a variety of shapes. They can also be made from a variety of materials like polymers, liposomes, and hydrogels, and can have different functional groups or ligands on the surface. All these attributes allow for targeted drug delivery and for the encapsulation of both hydrophobic and hydrophilic drugs. As seen here, with the drug encapsulating nanoparticle encapsulating the free drug, it allows for a protective coating to encapsulate this free drug, which bypasses the adverse side effects of drugs to the systemic organs, whereas these drugs can be toxic to the body when just being put in by themselves. The polymer we're choosing to use for our nanoparticle is polylactic co-glycolic acid, or PLGA. This polymer is biodegradable and biocompatible, which means it is safe to use within the body. It is also versatile encapsulation and can be modified for different disease treatment, which means a variety of drugs can be used within. Therefore, my study will use the PLGA nanoparticle as a carrier for LSF delivery. So current treatment therapies and route of administrations for RA do produce several adverse side effects. Therefore, an alternative, safe, and effective drug delivery system is desired. Alpha-fluoropane, a natural compound, is a proven non-inflammatory drug for use within the body. So therefore, the purpose of the study is to develop a nano drug delivery platform that enables the delivery of alpha-fluoropane for RA therapy. So we hypothesized that alpha-fluoropane loaded into polylactic co-glycolic nanoparticles will be effective for RA therapy. We plan to test this through two aims. The first being to synthesize and characterize the LSF-PLGA nanoparticle, and the second being to study the cytotoxic and anti-inflammatory effect of the nanoparticle. For the first aim, for the synthesis, we're using a water, oil, water emulsion, and solvent evaporation to create the PLGA and the LSF-PLGA nanoparticle. For the characterization, we're using the scanning electron microscope to look at the particle morphology, the dynamic light scattering machine to look at particle size and zeta potential, and the HPLC machine to look at the release kinetics and the loading efficiencies between PLGA and LSF. For the synthesis of LSF-PLGA, we first dissolved PLGA in DCM and then allowed LSF to stir within for 1 hour. We then dissolved PBS in the solution and then slowly pipetted the solution into PVA. We then sonicated the mixture, evaporated the organic solvent out, washed and centrifuged the nanoparticle twice, lyophilized at -80 degrees, and then stored the harvested nanoparticle at 4 degrees Celsius. We did take different methods to create this nanoparticle. The first method being using the original process. The second method, by allowing LSF to stir within for 5 hours and compared to 1 for the original method. And the third method, having lyophilized LSF straight to PBS and allowing that to stir overnight for 10 hours. These three methods did produce 3 different binding efficiencies, which we will examine on the later slide. As for the nanoparticle characterization, the nanoparticle was observed and added an SEM machine at Northwestern University. For the dynamic light scattering machine to look at size and zeta potential, we added 1 milliliter of the nanoparticle in deionized water. And for the HPLC machine, we added 4 milligrams of the LSF-PLGA nanoparticle that was dissolved in DCM the night before. As we can see from the image from the scanning electron microscope, we can see that there is no significant variation between the nanoparticle before the drug was loaded and the nanoparticle after the drug was loaded. We can also see from the results of the dynamic light scattering machine that the average size of just the PLGA nanoparticle by itself ranged around 150 nanometers. But after encapsulation, the size increased to around 200 to 230 nanometers. We can also see from a combined zeta potential, which is surface charge, of both PLGA and LSF-PLGA, that the zeta potential was between -9 millivolts and +20 millivolts. Since it is between -30 and +30, that we can see that this nanoparticle is in a stable dispersion, which means it is safe to use within the body. We can also see from our HPLC machine, which first we did load alpha-fluoropane in the machine to create a standard graph. This standard graph is also used in coordinates with this encapsulation efficiency equation to then create the binding efficiencies for all 3 of our nanoparticles created. From our first method, the binding efficiency was only around 30%. From our second method, the binding efficiency was around 50%. From our third method, with adding the lyophilized LSF to the PBS and then stirring overnight for 10 hours, we can see a binding efficiency of 80%. Since this method did have the highest binding efficiency, we used this nanoparticle for the rest of our experiments. We can also see from the 24-hour release study of the LSF PLGA nanoparticle that there was a consistent drug release from the PLGA nanoparticle compared to a burst release. We can see that 50% of the drug was released around 5 hours, and around 80% of the drug was released around 8 hours. On to our second aim, we used a raw mouse macrophage cell line to study cytotoxicity and the anti-inflammatory effect of the nanoparticle. To study cytotoxicity, we used an Alamar blue assay to look at the cell viability. And to study the anti-inflammatory effect, we used the CBA analysis to look at cytokine induction and cytokine reduction. To study cytotoxicity, we treated a 96-well plate and then treated with the LSF PLGA nanoparticle. After 24 hours, we treated with 10% Alamar blue and medium, and then red absorbance at 570 and 600 nanometers after 4 hours. As for the anti-inflammatory effect, we first treated with 500 nanograms of LPS, and after 24 hours, we then treated with our LSF PLGA nanoparticle. After another 24 hours, we then removed the supernatant, which we then used for our CBA analysis. From the results from our cytotoxicity study, we can see that L-ciforphane by itself does show increasing toxicity as the amount of the drug increases, compared to the PLGA nanoparticles by itself, which does not show any significant toxicity with cell viability remaining around 100%. Comparatively, the LSF PLGA nanoparticle does show cell viability remaining above and around 100% compared to LSF by itself with a very low concentration of cell viability. This shows that the LSF PLGA nanoparticle does not show toxic effect compared to the L-ciforphane drug by itself. We can also see from the LPS activation, the LPS activation induces cytokines so that we can later examine if the LSF PLGA nanoparticles will decrease the number of cytokines presented. So as we can see, there is a dose-dependent activation, whereas the amount of LPS induced increased the amount of cytokines produced. And 500 nanograms did show that the amount of cytokines produced was the highest, so we used 500 nanograms per mil for our CBA analysis. As for our CBA analysis, we can see that the activated macrophages by themselves have the highest induction of cytokines, as treated with the LPS. But once treated with the LSF PLGA nanoparticle, we can see a significant reduction of cytokine production, which shows that the LSF PLGA nanoparticle does have anti-inflammatory effect if it is reducing the amount of pro-inflammatory cytokines produced. On to our discussion, we can see that the standard size of a nanoparticle was around 200 to 220 nanometers, and that our surface potential was between -7 to +20 millivolts. Because of this, we can see it's just a stable dispersion of the LSF PLGA nanoparticles due to the electrostatic repulsion of the individual particles themselves. We can also see that there was no burst release of the drug observed in the initial hours, but there has to be a further analysis of the release kinetics in an acidic environment with a better understanding of LSF release at the inflammatory sites. We can also see that the inhibition of IL-6 and TNF-alpha cytokines by the activated macrophages also play key roles for RA therapy, which was observed in our study after the treatment of the LSF PLGA nanoparticle. So in conclusion, from the results of the study, our hypothesis is validated. The LSF dissolved in PBS, which was our third method, shows the highest binding efficiency of approximately 80% loading. We can also see that the LSF PLGA nanoparticles significantly reduce the toxicity of L-ciforphane. The IC50 of LSF by itself was observed to be around 5 microgram per mil. However, after the encapsulation of LSF, it did not invoke any significant toxicity up to 50% after up to 50 microgram per mil. We can also see from the anti-inflammatory studies of the nanoparticle showed a decreased levels of the IL-2, IL-6, and TNF-alpha cytokines compared to the activated macrophages by themselves. So from the results of this study, there's clear evidence of an anti-inflammatory response of LSF PLGA, showing the potential of this drug delivery system for rheumatoid arthritis. I would like to thank everyone on the slide, but specifically my mentor, Dr. Divya Bijukumar, and my co-mentors, Dr. Matthew and Dr. Kai, and Eddie. Thank you.
So that concludes our student presentations for today. At this time, I would request the judges to step out. to make the deliberations and select our final awardees. I would like to request Dr. John Rogers to come and hand out the participation certificates and also introduce the Dennis Klink Award.
We have a slide, I believe. So I want to thank everybody for great presentations, great interaction. You know, you have presence. And I think that's really important. I think you did a fantastic job with, you know, showing you're in control. You're the center of the room. Well done. I also want to thank Janaki and Amy, and I don't think we've met. I apologize. But a fantastic organization. Well done. So at this point, I thought there was a slide.
No, not for the participation certificates. I think there's a slide for Dennis Klink Award.
Okay, so what I wanted to do is ask Dr. Ramaswamy, who I don't see, and Janaki to join.
Oh, he's inside there, it seems.
Oh, okay. To join me in thanking, oh, sure. See, up here it sounds like I'm fine.
Right.
I can learn from the interns. Okay, can you hear me now? No? Okay, how about now? All right, good. It took a bit to warm up. Just like all of us, right?
There's no slide for the...
Okay, so I want to thank all of you. And Dr. Ramaswamy, if you could join me, I'd like to give certificates to all of the high school and college interns from the summer. And again, thank you for great presentations and for, you know, that passion. You know, I hope that this is just the beginning of your scientific career. You know, I remember when I was a summer intern, and that opportunity to get in the lab and see what all this theory is about is really is very important. It makes it real. So first, I'd like to ask Ruth to come up. And we'll ask you to stay up on stage because we're going to do a little photo op. And Varun. Thank you. Oh, you can't get away.
Sorry.
You already got away from me there. Let's see. Dhruv. Very good. I got you. Now I know what's going on. Branjay? And Shreya? There you go. Very good. Congratulations. Nice work. Yeah, there you go. Anastasia. Okay. Here, let's get that. And Maureen. Well done. And last but not least, Scott. Well, again, I want to thank all of you, the high schoolers and the college students, for all the great contributions to science. There we are.
Thank you.
All right. All right, so then I'm gonna, yep. Yeah, I'm gonna talk about Dennis Klink. So, you go ahead. Oh, actually, are they gonna stay up here, or is it sit down? Yeah, you guys, you're released. So, while the judges are out, I wanted to spend a little bit of time about what they're debating. So back in the 70s, a chemist joined the Pierce site, the Thermo site here in Rockford, and he had passion. He ate, drank, and slept science. Dennis Klink was so passionate about science that he inspired and really developed the mentality of how do we change the world. And so he had such an impact. Unfortunately, he passed away back in 2002. He was attending a conference and passed away while he was traveling. And so, you know, in remembrance of his legacy, we established an award to honor Dennis. And so every year we award the Dennis Klink Scholarship Award for, I believe there's a scholarship involved there, there's some money, for the high schooler and the top high schooler and top college presenter. And this is based on the judges' assessment this morning from their presentations and Q&A session, as well as today's presentation, and I think perhaps the posters as well. But I'm not sure, given this year with COVID. And so we select one high school student and one college student by the panel by the judges. And really, this is meant to encourage that passion and encourage, you know, a lifelong dedication to learning. No matter what your career is, you never stop learning. So I also want to thank some of our judges. So I'm waiting for them to come in the room. So, oh, okay, so I don't need to kill more time. Good. So first of all, I'd like to introduce the judges. So Deborah Breiter, and so she's the Dean of College of Science at Rockford University. Dr. Ryan Baumgarten, so he manages research and development efforts at the Thermo Fisher site here in Rockford. Aram Raja is one of our staff scientists, a chemist, again at the Rockford site. Khalifa is a director of the master's program here at the School of Medicine. And Vishal Khatri is a research specialist. So I'm looking for them to come in and share with us, you know, drum roll, decision on the Dennis Klink Award. Very good. Now, in the past, we had a visitor from Japan that used to come every year to participate as a judge. And this year, I think due to COVID and other issues, he wasn't able to attend. And so we had to mix it up a little bit. So, but oh, he is? Oh, good, good. Well, if he is here, hello again. Oh, they went out for coffee. Good, good. Okay. Do you still have the microphone? I was going to ask the interns, not to put anybody on the spot, but I'd love to hear your, what did you love the most? What did you hate the most? I don't know. Give me something that comes top of mind from your experience, your intern program this year?
I really love working with my lab partner and my mentors. It really made this experience really fun for me. Like the amount of time we had for leisure time without like stressing too much about our work was really nice. Just getting that hands-on experience was really beneficial to me. I really appreciate like the experience and the opportunity I got to work in this lab as well.
Good, good. The word I heard most was fun. You know, it's great to get in the lab and enjoy what you do. It makes it easy to go to work every day, and it inspires you to do better. So, anyone else? Anna?
So I would totally agree with that. I definitely have sensed collaboration at Thermo Fisher Scientific that really kind of surprised me at first. But the fact that you can build on a team and really explore new options, and yeah, the joy of discovery of like, hey, something worked really well. So that was something that has really stood out to me, and I'm really going to miss those folks that I worked with this summer.
Good.
Yep, definitely the people. I think it was a really nice opportunity to be able to work one-on-one.
Can't hear you.
Oops, sorry. Definitely the people. It was a really great opportunity to be able to work one-on-one with so many great scientists and really get a lot of advice and just technical information.
Yeah, I would agree with what the other intern said. I think one of the best things about working at Thermo Fisher Scientific is that there are a lot of really, really intelligent people that would love to just talk to me about their projects and what they've been doing, as well as hear as what I've been doing. And I think it's been really nice to just learn more about what an industry is like rather than academics. I think that's something really rewarding as well.
Very good, very good. Any other comments? Oh, look at him.
I think my favorite part was understanding how research is a confluence of science. It's where a physics major and a chemistry major can come together and create something totally cool and innovative.
That's right. That's right. Very good. Yeah, I took a new role like a year and a half, 2 years ago, trying to consolidate or better align chemistry efforts across a range of chemists at different sites. And one of the real opportunities there is to identify best practices and share ideas and give people new perspectives and greater visibility. And internship programs like this, give us visibility to high schoolers and college students, right? And we have a number of people number of scientists at the Rockford site, I know, that actually we were summer interns back in the day, and came back to continue, maybe as a real employee instead of a short-term summer, it had an impression. So we use this not only to develop new scientists, to develop staff and new leaders, but also to develop the next generation of employees. So what's going on? Do they have like a 5-way tie?
They're having difficulty making their final decisions.
What's going on?
That
Oh, okay. Okay, very good. Well, that's a good thing, right? Well, I don't envy their decision.
Yeah.
Okay, good, good. All right, well, drum roll, I guess. Yeah. I was going to ask some of the high schoolers some questions, but I'm not going to put you on the spot. Did you have a question? Oh. I'm sorry? Yeah. Yeah. I don't remember ever seeing them take so long to make a decision, so. Still working at it. Well, I think that that's a very worthy. Some people, they just step right up and, they take it for the team. They take it all for the team. Good, good. Well, earlier this week we had a picnic. And it was great to meet all the interns. I think, I know for myself, I only saw 3 or 4 people at the Rockford site. I didn't realize how many interns we had. It was great to get everybody together, but I'm not familiar with other events that were held. I came back into town just for you. But I don't know if anyone can comment on, maybe you could comment on some of the other events that they were involved in over the summer? Yeah. Well, it's a little different from previous years. Yeah. Okay. Oh, good. All right. There they come. So again, I introduced our judges, and I'm going to ask the judge, at least one judge, to come up, and okay, you'll present the awards. So again, yeah. So come on up, Dr. Breiter, Dr. Baumgarten, Dr. Raja, Sadiq, and Khatri, and I hand it off to you. You guys are making me. You're pressuring me here. Okay. All right, here, I'll give you this. Sure.
All right, so thank you for your patience. We were back there deliberating a lot because all the students did an amazing job. We're impressed year on year at how good the presentations are, how composed you are, how much knowledge you gained in only doing 8, maybe 10 weeks worth of research. I know all the experiments didn't work the first time, and there's multiple repeats to get the data you got, and some amazing science was presented here today. So please congratulate all the interns for their presentations today. And as I mentioned, we'll be presenting the Dennis Clake Award for the best intern presentation and interview from the committee. And again, the voting came down to less than 1 point between the top 2 people. So it's my pleasure to announce for the high school student award that Dhruv Ramaswamy has won the internship. Oh, congratulations. All right. And for the College Intern Award, it's Anastasia Klenke. Yep, and I guess we'll do a photo with the judges, probably with whoever's taking photos. But I guess the rest of us will have is there a reception, Janaki? What's next? Yep. So I guess you want to conclude the ceremony or just let me do it? We're good to go. Thank you all for coming, and I guess that's the end of the intern presentations.